And we did that. We converted that carboxylic acid into the acid chloride and attached an amine to form an amide bond. And we showed that we did it quantitatively. And really what this gives us is an opportunity to control the size and the chemistry of the pore, because now we can make the pore hydrophilic or hydrophobic, charged, positively charged, negatively charged. We can attach something to it, and we envision attaching lids that can be stimuli responsive. So it could be a lid that opens up when you change the pH or shine light on it, or it could be chemical stimulation. Suppose that there is binding event that opens or closes the pores. So all this is quite exciting. And of course, since we started working with all these colors, we decided to expand and get as many combinations as we could. And these are electron micrographs of these polymer capsules. Transmission electron micrographs, and you see the scale bar is about 100 nanometers, so we can create very small capsules. This is a scanning electron micrograph, and of course it's at the limit of its resolution, so the picture is a little hazy. But you can see clusters of these capsules, like grapes. And that's important to us because these capsules were dried after solvent exchange. They were dried, and then we simply took them. And it was good to see that the material, which has only 1 nanometer thickness in this capsule wall, can survive regular treatment. You don't really need to do anything special to it. You can dry it up, and it still retains its shape. So you probably cannot see what is written here, but it says, University of Memphis scientists created the world's thinnest membrane, because it is a membrane. We demonstrated good permeability control, and the thickness is smaller than the biological membrane. So the biological membrane is 5 nanometers thick. The synthetic membrane is about 1 nanometer thick. It's probably hard to get much thinner than that because it's only 1 or 2 molecules cross-linked. So where can we go from there? Since we have a material which is about a nanometer in thickness, and we can control the pore size and pore chemistry, there are several opportunities that we can pursue and that have interesting implications. Of course, ultrafast dialysis, so 5-minute dialysis and no more long coffee breaks for molecular biologists. There is a very interesting application of pores in DNA sequences. In DNA sequencing, if you take a single-stranded DNA and pass it through a pore, then each base, having slightly different size, would modulate the ion conductivity across the pore. And if you're lucky, if you have a thin enough material with precisely controlled pore size, then when you pass that DNA strand, that maybe you would be able to record the ion conductance, and then that would really tell you what is the sequence. And it's an exciting way to sequence DNA, and of course new materials are important to approach that. Nanoreactors is a very interesting application where we can entrap catalysts, either enzymes or organometallic catalysts inside the capsules, and allow for homogeneous environment and let substrates to get in, get converted, products can get out. This is something that we will be presenting next week at the Global Venture Challenge. I'm going with a team of graduate students to Oak Ridge, and we'll see how that technology will score there. And when we put these membranes onto an electrode surface, we create new sensors, because if we can control the perm selectivity, if we control the thickness of that layer, and we create recognition sites in the pores, then we can achieve either new sensors for medical sensing, or also we could use that as a platform for depositing stimuli responsive materials and get to a new generation of sensing based on environmental changes. So there are many interesting opportunities that we can pursue having these materials in hands. And of course, something similar to that, capsules containing selective pores can be used for drug delivery or drug release applications, because you can envision these are containers that have fairly good capacity, very thin shell, and when we learn how to regulate the transport by opening and closing the lids, then that would give us the opportunity to do drug release. And we have another application related to capsules that we didn't talk about today, but it's dealing with the combination of diagnostic and therapeutic applications. We could load capsules with contrasting agents and bind them selectively to a certain tissue. And that's something that we are pursuing in collaboration with Karen Hasty from UT. It's on arthritis treatment. So that's about it about our nanoporous membranes.